Hi there, Dr. Hudson. I'm Matthew Yacht, and this is my membrane concept map. I have divided the concept map into 3 subcategories. Wrapped around in the green we have components, and then over here in the blue we have like general function of membranes, and then over here wrapped around in the pink we have models of the plasma membrane. So we'll start over here with my components. It's divvied down here to amphipathic molecules and membrane proteins, and then also down here we have components continued. We have other significant molecules and channels and pores. We'll start right here with amphipathic molecules. Right here we have the phospholipid bilayer, which is made up of a hydrophilic head and a hydrophilic tail, and the hydrophilic tail has both unsaturated fatty acids and saturated fatty acids. Unsaturated fatty acids are fatty acids where the carbons are do not have their maximum number of hydrogens, and then saturated fatty acids are where they do. And over here also another amphipathic molecule is sterols. And then we'll move on to the membrane proteins, and the main 4 are trans, anchor, enzyme, and receptor. And then over here we have forms, which is how like the proteins can be attached to the plasma membrane. Lipid linked, we have protein attached to another, which is essentially proteins just bonding non-covalently with another protein that's attached to the plasma membrane. And then monolayer associated, that's where it's attached to one layer. And then over here, along with membrane proteins, we have integral versus peripheral membrane proteins. And those are, that brings up the topic of if you want to study the proteins in the membrane. Peripheral are generally easy to get out because you can take them out through like easy means, but integrals are in the plasma membrane, so they need to be taken out with detergent, which is other significant molecule. Another significant molecule is glycoprotein and glycolipid. And then over here we have our next category, channels and pores. We have membrane channels and ion channels. And ion channels are typically associated with transport proteins and stuff. And then membrane channels are just channels within the plasma membrane, within the phospholipid bilayer. And then they both have selected permeability. And if ion channel has a selective permeability, one example is a gated ion channel, which brings me over here to my osmosis side. Osmosis is driven by diffusion, which is just particles randomly spreading out through thermally driven movements. And usually osmosis is caused by a hypotonic or a hypertonic concentration, either outside or inside the plasma membrane. And osmosis wants to drive either these hypertonic or hypotonic solutions to be isotonic, which the concentration on both sides of the semi-permeable permeable layer is equal. And over here we have active transports and passive transports. And active transports are uphill reactions, so they are energetically unfavorable. So we say they go against the electrochemical gradient, which is the driving force of osmosis in the plasma membrane. And then we also have passive transport, which are downhill reactions, so we say it follows the electrochemical gradient. And a form of passive transport is the channel mediated passive transport, which relates back to our components over here, since that is performed by the gated ion channel. And finally over here we have our models of plasma membrane. We have One of them is called the Sandwich Model by Dason and Daniele. And then over here at the very bottom we have Fluid Mosaic Model by Singer and Nicholson. So that is it for my plasma membrane concept map. I hope you enjoyed. Thank you.